Assalamualaikum and good morning everyone. So today we're going to discuss about the cell wall, right? Which I should have included in the previous video. Tapi tidak kan. Okay, cell wall and then the transportation of substances across the cell membrane. Okay, macam mana apa ni? Bahan-bahan okay, dia dibawa masuk ataupun keluar daripada cell membrane itu. Okay, so all those you need to know. Okay, students. Right, so first things first about cell wall, right? So cell wall, okay, is a carbohydrate layer of cellulose, okay, found outside the plasma membrane of plant cells. Okay, there are two types of cell wall. Okay, the primary cell wall and the secondary cell wall. So now, first things first, let's dig into the primary cell wall. Okay, so this primary cell wall, they are found in young cells. Okay, young cells and cells that are not highly differentiated. Okay, example like the meristem, okay, the parenchyma and collenchyma. Okay, and then this primary cell wall has thin layer of randomly arranged microfibrils. Okay, you can see these microfibrils ini hijau ni, right? Thin layer of randomly arranged. Boleh nampakkan dia secara random saja. Ada yang macam gini, ada yang sengit sikit, ada lagi sengit, ada yang tegak, ada yang melintang. Okay, thin layer of randomly arranged microfibrils in an amorphous matrix. And then this amorphous matrix, yang ini amorphous matrix ni, okay, consists of pectin and hemicellulose. Okay, boleh nampak ini pectin yang purple ni, and then hijau ni hemicellulose. So matrix ini ada banyak pectin dengan hemicellulose. Okay, so if you're wondering what is the component of these microfibrils, right? These microfibrils, okay, kalau kamu at cellular level, they consist of chains of cellulose. So these chains of cellulose made up this microfibril, and this microfibril made up the fibril, right? Ini adalah fibril. Okay. Now, other characteristic of this primary cell wall, alright, the outer layer of the cell wall has middle lamella, or also known as pectin layer. Okay, kalau kamu tengok dalam gambar raja ini, okay, this is the primary cell wall yang kuning ini. So between one primary cell wall with another primary cell wall, okay, there is a middle lamella. Okay. Next, this primary cell wall, okay, they are strong, elastic and extensible. Okay, extensible. And this allow parenchyma cells to become turgid that support the plant cell. Okay, or eventually support the plant, give turgidity to the plant. Now, this primary cell wall, they are usually perforated with plasmodesma for transport of substances between cells. Now, class, the region of this primary cell wall, okay, this is the primary cell wall. If you look from this diagram, this is the middle lamella. Okay, they have a region where it is thinner compared with the other region of the primary cell wall. That thinner region, alright, of this primary cell wall is known as pit. Okay, pit. And this pit has plasmodesma or plasmodesmata, being plural. Okay. So now class, the point of today's talk yang ini, kamu mesti boleh nampak, you must be able to identify, okay, where is the middle lamella, which is this one obviously, and then this is the primary cell wall, and then secondary cell wall. Okay, after secondary cell wall, di sini ni ialah plasma membrane. Okay, in a clearer diagram, ini ialah middle lamella, boleh nampak Okay, tengah-tengah ni. Yang ini middle lamella. Untuk yang apa ini? Primary cell wall, cell 1. Ini middle lamella untuk primary cell wall, cell 2. Okay, ini lagi jelas. Kalau ini kamu tengok middle lamella ni satu saja. Actually dia ada dua masing-masing. Primary cell wall yang ini ada middle lamella dia. Primary cell wall yang cell 2 ini dia ada dia punya own middle lamella. Okay. So okay, next. Primary cell wall, they are porous. Okay, porous means this primary cell wall enables water to be transported via apoplast pathway. Apoplast pathway ni you learn in detail in semester 2. Okay, now if you're wondering what is apoplast pathway, by the way, it's a pathway where most water and mineral ions diffuse along the porous adjacent cell wall. Lepas ni kamu boleh nampak how the water and mineral ions it diffuse from one plant cell to the other plant cell via cell wall. Okay, or to be more precisely, the primary cell wall. Okay, nampak Okay, now the secondary cell wall. Okay, it's harder. Alright, secondary cell wall is harder and thicker layer of cell wall is formed between the plasma membrane and the primary cell wall. So di antara plasma membrane dengan primary cell wall ialah secondary cell wall. Kalau daripada gambar aja ini, from this diagram, alright, yang ini, this is the primary cell wall. Okay, boleh nampak pun this is the primary cell wall. Okay. And then mana secondary cell wall? Yes, yang ini adalah secondary cell wall. Yang lightly stain ini adalah secondary cell wall. Tapi yang sini pula ini adalah secondary cell wall. Okay, boleh nampak dia thicker. Primary cell wall ni thicker compare dengan primary cell wall. Secondary cell wall ialah thicker. Pasal cikgu cakap ni. Secondary cell wall. Okay, the secondary cell wall is thicker if you compare with the primary cell wall. Okay, where is the cell membrane? Ini ialah cell membrane. This is the cell membrane, right? Boleh nampak Okay. And then this is the plant cell cytoplasm. Okay. Yang putih ni ialah the plant cell vacuole. Okay. Mana nukleus? Nukleus yang hitam kecil ni sini. This is the nucleus. Can you see? Right. Okay. Boleh nampak secondary cell wall. Okay, now the secondary cell wall made from regularly arranged, microfibrils or macrofibrils in layers of parallel rows which are perpendicular to the upper or lower layers. Okay, you can see how these microfibrils or macrofibrils, they are being arranged perpendicularly, right? Now, the matrix of this secondary cell wall, as you can see from here, ini secondary cell wall, ini matrix ini, right? Filled with lignin. Okay, so diagram ini lagi jelas. Okay, you can see this secondary cell wall. Okay, the matrix is filled with lignin, right? So this lignin form the hard impervious layer. Alright, impervious or impermeable layer. So meaning to say, in the secondary cell wall, water, okay, cannot pass through. Okay, and this secondary cell wall, they are not extensible, right? If you compare with the primary cell wall, they are apparently extensible. So yang secondary cell wall, they are not extensible. So mostly the secondary cell wall, okay, they can be found in supporting tissues as well as water conducting cells. So kalau dalam gambar aja ini, this is the plasmodesma, alright, or plasmodesmata. Ada banyak. Okay, ini ialah cell membrane. Okay, cell membrane. I think dari sini ke sini, right? Sorry, okay, inilah cell membrane, and then kita ada secondary cell wall yang ini, okay. Yang nipis ini ialah primary cell wall, sangat nipis ni, sangat nipis, okay. Ini ialah primary cell wall. Then kita ada middle lamella di sini, right? So summary, okay. So students, at this point you must be able to differentiate between primary cell wall and secondary cell wall, okay. So these are the differences between the primary cell wall with the secondary cell wall, okay. Now let's continue with movement of substances across cell membrane, okay. So these substances, they move across the cell membrane in 4 ways, okay, 4 cara. So first is passive transport, and this passive transport, okay, the substances, they are being moved, okay, follow the concentration gradient, meaning to say from a higher concentration region of that substance will move to a region that has a lower concentration of that substance. For example, like water diffuse from a higher concentration region to a lower water concentration region, okay, dari kawasan yang berkepekatan tinggi ke kawasan yang berkepekatan rendah. Now there are 3 types of passive transports, right? Simple diffusion, facilitated diffusion of facilitated transports, okay, and osmosis. Okay, semua ni berbeza, but all these 3 are passive transport. And then from this diagram here you can see this is simple diffusion, okay, where the molecules or substance just pass through the phospholipid bilayer, okay. And then we have facilitated transport, okay, or facilitated diffusion that involves channel protein and carrier protein, or channel protein or carrier protein, okay. And then we have osmosis that involves aquaporins as well as semi-permeable membrane, okay. Okay, aquaporins dia di sini ya. Now kelas, simple diffusion and osmosis ada beza dia. Kalau simple diffusion ini, there are many examples of simple diffusion, but one that is very common is the simple diffusion of water. So the water will just diffuse from one point to another point, right? Or from one point that is higher in concentrated water molecules to a point where the water molecule concentration is lower, okay. But when it comes to osmosis, it is the movement, or sorry, osmosis is the diffusion of water molecules, right, that involves semi-permeable membrane. Yang simple diffusion ini tidak melibatkan semi-permeable membrane, okay. Now active transport, this is the transportation of substances that is against the concentration gradient, okay. And this active transport, right, involve the proton pump and coupled transport or co-transport, okay. So kalau kamu jumpa co-transport, other name for co-transport is coupled transport, okay. Now the proton pump, okay, sorry, yang ni ada pembetulan sikit ni. Proton pump, dia proton pump, can be proton pump, okay, pump pula, or sodium or potassium ion pump, okay. Alright, dan ini active transport, okay. You can see how this substance is being pumped, alright. to a region with this is lower concentration region, this is higher concentration region. So the substance here is being pumped out, okay, from lower concentration to higher concentration region. Now, endocytosis, okay, it's the third way how the substance can be moved across cell membrane, all right? And this endocytosis needs energy. Okay, examples of endocytosis are phagocytosis, pinocytosis, and receptor mediated endocytosis. Okay, and the fourth is exocytosis. Okay, and exocytosis needs energy. So number 2, 3, and 4 needs energy. Only number 1, passive transport, does not need any energy. Now let's get into a detail into each one of these example of passive transport, active transport, endocytosis, and exocytosis. Okay, now class, endocytosis and exocytosis are also known as bulk transports. Okay, so these are the summary, right, of the which movement needs energy, which movement does not need energy. Okay, so energy required for the active and bulk transport. Diffusion and facilitated transport does not need any energy. And then the energy is in the form of ATP, okay, adenosine triphosphate. Now, passive transports, okay, transport of substances without the use of energy. Okay, characteristic follows concentration gradient from region of high solid concentration to low solute concentration. Okay, equilibrium is reached. Equilibrium ialah keseimbangan. Okay, is reached when solid concentration on both sides of the membrane are equal. Okay, so the amount of solid molecules moving in and out via the membrane or via the cell membrane are the same. No net movements of solids. Okay, no net movement of solid meaning to say, okay, bukannya tidak pergerakan bahan itu from region A to region B. Okay, no net movement means they are movement of that solute from region A to B and B to A, but in equal numbers. Okay, that's why I mentioned here the amount of solid molecules moving in and out via the plasma membrane or the cell membrane are the same, sama. Okay, and that we call no net movement. Usually students, you tend to think no net movement meaning to say tiada pergerakan solid itu, langsung tiada pergerakan. Salah. Alright, sebenarnya ada, tapi dia equally the same from both sides. Now, this passive transport may require a specific transport protein, right? May require a specific transport protein to transport specific solute across the membrane. Okay, so that's why yang passive transport yang may require a specific transport protein ini dia dikategorize sebagai facilitated diffusion, diffusion yang dibantu, right? Facilitated diffusion. Kalau tidak perlu specific transport protein ini, dia ialah simple diffusion saja dan juga osmosis, right? So 3 types of passive transport: simple diffusion, facilitated diffusion or facilitated transport, and osmosis, right? So now simple diffusion is the movement of substances from a region of high concentration to a region of low concentration, okay, without passing through the semi-permeable membrane. Driven by kinetic energy and chance movement due to the molecules always in a state of movement and moving away from high concentration. Ini seperti dibincangkan dalam theory of kinetics. Okay, so if you are wondering why the molecules can perform simple diffusion, it is because those molecules are driven by their kinetic energy. Okay, from their kinetic energy there is a chance movement. Now, examples of solid that perform simple diffusion, Okay, they are gases and lipid soluble substances. All right. Now, small hydrophobic, okay, and uncharged polar molecules. Sorry class, let me rephrase that. Okay, example, solids that perform simple diffusion are gases, okay, gases and lipid soluble substances, right? Then small hydrophobic and uncharged polar molecules, right? These four category, four types of solids: gases, okay, lipid soluble substances, and then small hydrophobic substances. Example of the small hydrophobic substances, they are the oxygen, okay, carbon dioxide, nitrogen, benzene, okay, and then uncharged polar molecules like water, ethanol, and glycerol. Okay, so now example of this simple diffusion is like high concentration of glycerol outside the cell will make the glycerol easily slip through the phospholipid bilayer into the cell. Okay, ini saya tambah sikit sini. Sample. Betul. Alright class, ini lebih clear. Right, so example of solids that perform the simple diffusion are gases, lipid soluble substances, okay, and then small hydrophobic substances like water, CO2, nitrogen, benzene, okay, and then uncharged polar molecules. Okay, next, factors affecting simple diffusion. Okay, one is the concentration gradient, obviously. Okay, so directly concentration gradient is directly proportional to the rate of diffusion. Meaning to say, the bigger the concentration gradient, the bigger the rate of diffusion. Lagi besar concentration gradient itu, lagi cepatlah rate of diffusion. Okay, temperature, so directly proportional to the rate of diffusion. The higher the temperature, the higher the rate of diffusion. Then surface area of membrane, okay, betul juga, directly proportional to the rate of diffusion. Lagi besar surface area membrane itu, lagi cepatlah rate of diffusion. Okay. And then the size of diffusing molecule lah, ini inversely proportional to the rate of diffusion. Lagi besar molekul itu, lagi perlahan lah, ataupun lagi kecil rate of diffusion. Okay. Polarity of diffusing molecule, ini pun sama juga, inversely proportional to the rate of diffusion. Example, non-polar molecules easier to diffuse. So rate of diffusion increases with low degree of polarity. Okay. Okay, yang F, diffusion distance. Yang ini pun inversely proportional to the rate of diffusion. Lagi pendek, okay, the shorter the diffusion distance, the greater the rate of diffusion. Okay, the shorter the diffusion distance, the greater the rate of diffusion. Okay, facilitated diffusion, right? Yang ini, as what I've mentioned earlier, it does not need energy, but it requires the potential energy of the concentration gradient. The same goes with the diffusion, the simple diffusion. Okay. So, facilitated diffusion, diffusion that facilitated by carrier proteins. Okay. So, movement of hydrophilic molecules. Facilitated diffusion is the movement of hydrophilic molecules or ions across the cell membrane via transport proteins or carrier proteins and channel protein as well. Okay, that allows the concentration, that follows the concentration gradient. Okay. All right. Before I continue, class, carrier proteins and channel proteins dia sama atau bentuk dia. Kalau kamu tengok di sini, right? This is a channel protein. Okay? Yang integral protein itu, integral plasma membrane protein itu, di tengah-tengah dia ada lubang, ada channel. So the substance, the solid just pass through. Okay, through the channel. So nama dia ialah channel protein. Right? Carrier protein dia berlainan sedikit macam ni. Okay, it takes in the substance and takes in the substance, masuk sini, takes in, and then it remove the substance inside the cell. Okay, carrier protein dia lain sikit. Now, direction of net movements determined by the site of membrane that has higher solid concentration. Alright, if this region has higher solid concentration, so the movement will be from this region to this region, vice versa. Lemak, low battery pula. Okay. Alright, so before I proceed, okay, tak apalah, proceed dulu. Because I wanted to talk about osmosis actually. Okay, never mind. Alright. Equilibrium is reached when concentration of solid on both sides of the membrane are the same. There's no net movement of solid. Again, class, what does it mean by no net movement of solid? Okay. Maksud no net movement of solid ialah pergerakan solid from this region to this region and from this region to this region equally the same. Alright, masih ada pergerakan solid tetapi sama rata from this region to this region and this region to this region. Right, there is no net movement. Okay, contradictory to what you might thought that no net movement means langsung tiada pergerakan. That is wrong. Okay, the transport protein can be saturated by solutes. Alright, the transport protein can be saturated by solutes when all of the transport proteins are in use, and any increase in the solid concentration does not increase the rate of diffusion. Okay, so facilitated diffusion or facilitated transport by a carrier protein can speed the rate, alright, at which a solid crosses the plasma membrane toward a lower concentration. Okay, note that the carrier protein undergoes a change in shape as it moves as solid across the membrane. So, yang carrier protein ini dia akan bertukar bentuk dia bila dia pindahkan solid from one point to the other point. Now, example of substances, okay, that can be moved by this facilitated diffusion, right? Okay, one example is for small water soluble substances. Okay. Now, one example of small water-soluble substances is sodium ions. So, sodium ions diffuse into the nerve cell. Okay. Boleh nampak sodium ion diffuse into the nerve cell. Okay. Or potassium ions out of the nerve cell when there is no impulse via carrier proteins. The nerve cell. Okay, class. So this is one example of carrier protein, right? These are the example of carrier protein. Remember, just now I mentioned that in this facilitated diffusion, it involves either carrier protein or channel protein. Okay, ini carrier protein. Okay, tengok bentuk dia berubah. Okay, ada dua gambar aja sini. We go to this one first, right? This diagram here. It shows you these potassium ions, okay, move out or diffuse out of the nerve cell, okay, by the carrier protein. So if you notice the shape of this carrier protein, it changes. Initially, it was like this. Then when this carrier protein transport the potassium ion, the shape changes to this. Boleh nampak dua shape ni tiada sama, dia berubah, sedikit berubah. Okay, the same goes here. When this substance here, all right, for example, sodium ions, all right, when it being transported or diffused into the cytoplasm, okay, look at the shape of this transport protein. Okay, this carrier protein, it's slightly changed. All right, so carrier proteins bind specific, okay, to the molecules that they transport. You can see in this diagram here, this carrier protein binds to the specific molecule it wants to transport. Okay, the molecule concentration is higher outside the cell, as you can see from here. So molecules bind to the carrier protein on the outside. So the carrier's shape changes. You can see it changes from here to here, okay, to here, allowing the molecule to cross the membrane. Right, this is reversible. So net movement continues until the concentration is equal on both sides of the membrane. Okay, so maksudnya benda ni akan berterusan, either dia masuk ataupun dia keluar nanti bila sudah equilibrium. Okay, again another example of carrier protein. You can see this carrier protein, how it changes its shape daripada V kepada inverted V. Okay, this is GLUT1. Okay, this is GLUT1 protein carrier. Okay, it is a type of glucose transporter. Okay, and this can be found on your liver cell. Right, this GLUT transporter transport glucose into your liver cell. Okay, why into your liver cell? You will learn later in the glycolysis. All right. Now, another example is glucose. Okay, glucose diffusing into and out of the red blood cell via channel protein. Just now I talk about the carrier protein. Now about channel protein. Nampak beza dia? Channel protein dengan carrier protein tadi. So glucose, okay, they are hydrophilic and too big to diffuse across the phospholipid layer. Okay, you see, this is glucose. They are just too big to diffuse across the phospholipid bilayer. So they need assistance to cross the phospholipid bilayer. Okay. Now, on the left of the concentration, all right, on the left, the concentration is higher outside the cell. So the ions will move into the cell. And on the right, you can see here, the situation is reversed, where the concentration of the solute is higher inside the cell. So the ions will move out from the cell. So this diffusion will continue until the concentration is equal on both sides of the membrane. At this point, ions continue to cross the membrane in both directions, right? But there is no net movement in either direction. Okay, there is no net movement in either direction. All right, now let's continue with osmosis. Okay, so it's the movement or diffusion of water molecules from a region of high water potential, all right, to a region of low water potential through a semi-permeable membrane. So now class, now for diffusion, okay, it can involve water. All right, it may involve water for diffusion, but if the diffusion involves water and a semi-permeable membrane, all right, you don't call it diffusion, you call it osmosis. Okay, sorry, the movement of water, all right, the movement of water, you can say that this water molecules, it moves from one point to a point. Now, if the movement of the water from one point to one point involves a semi-permeable membrane, that is known as osmosis. Okay, if the movement of the water from one point to another point without involving any semi-permeable membrane, okay, if it follows concentration gradient, then that is just a simple diffusion. Boleh beza diffusion dengan osmosis. Diffusion ini dia boleh melibatkan bahan-bahan lain, tak semestinya water. Tak semestinya water kalau diffusion. Okay, boleh melibatkan bahan-bahan lain dan tidak semestinya water. But osmosis, it must involve movement of water as well as a semi-permeable membrane, right? And then the movement of water will be from a region of high water potential or to a region of a lower water potential. Okay, water potential ni maksud dia ialah concentration of water molecules. Because in water molecules, we don't say that they are being concentrated or lower concentrated. So we refer this condition as high water potential and low water potential. Okay, and water potential is being symbolized as phi. Okay, a trident. Okay, phi or trident. So water potential of pure water is zero. It is always zero. Okay, so meaning to say that environment has many water molecule. All right. If it's positive, then meaning to say that environment has more water molecules. Any solid solution, okay, water potential has negative value. So meaning to say any container, for example, a container or an environment that has solutes other than water, right? The water potential for that environment would be a negative value. Boleh ya? Contohnya satu gelas kamu ada dua gelas, okay, you have ada dua gelas or two beakers. You have two beakers. Beaker A you have only water, pure water. Beaker B you have water, then you mix with a pinch of salt. So the water potential of the beaker A, the beaker that has only pure water, will be zero. And then the water potential for the beaker B, right, the beaker B that has a pinch of solute, a pinch of salt, will be a negative value. Right? What will happen if you immerse a cell into a concentrated sugar solution? Okay, obviously you have learned this before. All right, water will diffuse, all right, out from the cell, okay, via osmosis, causing the cell to plasmolyze or shrink. So equilibrium is reached when water potential on both sides of the membrane are equal. Okay, so in living cells, all right, water flow in living cells is facilitated by aquaporins, which are specialized channels for water. Okay, and these aquaporins can be found abundantly in your nephrons. Okay, your distal convoluted tubule in your nephrons. Then these nephrons can be found in your kidney. Okay, you learn in more detail about these aquaporins in the second semester, or you can search in my channel in the kidney chapter. Okay. All right. So by this point, you should be able to identify. All right. What is A, B, C, and D? Okay, which one is simple diffusion, which one is a facilitated diffusion, and which one is a carrier? Which one is a transportation that involves a channel protein, and which one involves a carrier protein? Okay, so we discuss this in our Google Meet. So now, active transports. Okay, it is the transport of substances across membrane against the concentration gradient. Okay, which requires either a co-transport or proton pump and ATP. Okay, so untuk active transport dia melibatkan either co-transport protein ataupun proton pump protein dan juga ATP. Alright, so each co-transport protein or proton pump protein is specific for one type of substance, and some may transport, some refer to this proton pump or co-transport pump may transport more than one type of substances. Okay. Now class, selain proton pump, okay, cikgu nak tambah sini sikit. Which requires either co-transport, okay, or proton pump ataupun sodium ion pump, okay, sodium ion ataupun potassium ion pump. Okay. Now, the core transport of protein pump has a receptor site that allows substance recognition and binding. Maksudnya contoh macam ni lah. This is a sodium ion, right? This is a sodium ion pump. Okay, the sodium ion pump has a receptor site here. Okay, that allows the sodium ion pump to recognize sodium ion. Okay, because the sodium ion pump only pump or transport sodium ion into the cell or out from the cell. Okay, so now rate of active transport increases when ATP production is increased by either increasing the temperature and oxygen concentration or increasing the numbers of mitochondria. And you know mitochondria, they supply ATP, right? And mitochondria is the only organelles in our body that are able to generate ATP. Okay, and then another factor is cells with larger surface area have higher rate of active transport. Okay, because when the cell surface area is larger, all right, that cells apparently has more this co-transport protein or proton pump or sodium ion pump. Okay. Now, examples of this active transport, right? Active absorption of mineral ions into the root hair cells via co-transport protein. Okay. And then active absorption of glucose into the epithelial cells of the small intestine. Okay. Then sodium potassium pump that pump sodium and potassium into and out from the nerve cells to maintain a potential difference. This potential difference you learn in detail in second semester, or you can search in my channel. It's there. Okay, in the action potential topic. Okay, now more detail about the sodium potassium pump. All right. Or proton pump mechanism. Now, class, when it comes to proton pump, it involves the pump of or the transportation of hydrogen ions. Okay, and in biology, hydrogen ions, they are also known as proton. Okay, so whenever you see the word proton in biology, it is more likely referring to the hydrogen ions. Okay, so now to cut short, okay, about this on this mechanism of this sodium potassium pump. Okay, this sodium potassium pump mechanism. So what happens? All right, this sodium potassium pump, usually it will transport 3 sodium ions, okay, out from the exoplasm or nerve cell, and this sodium potassium ion pump will transport 2 potassium ion into the nerve cell. Okay, maksudnya 3 sodium ion dipump keluar dan 2 potassium ion dipump masuk ke dalam nerve cell. Okay, usually this is the duty of the sodium potassium ion pump or sodium potassium pump. I think it's better if we use the word here sodium potassium ion pump. Right. Kalau proton pump mechanism pula, alright, usually it's proton pump mechanism, it will pump proton into the cell or pump the proton out from the cell, right, or hydrogen ions. Now, co-transport on the other hand, okay, you can see how a co-transporter is, or co-transport protein. Very much different compared with the proton pump or the sodium potassium pump. Now, not to make you confused, okay, students, sodium potassium pump ini is a one type of pump, alright, that pump 3 sodium ion out from the nerve cell and pump 2 potassium ions into the nerve cell. Itu sodium potassium ion pump. Sodium ion pump pula, okay, dia akan pump sodium ion saja, either into or out from the cell. The same goes for the potassium ion pump that pumps only potassium ions only, either into or out from the cell. Okay. Now, co-transport protein, all right, is an active transporter driven by a concentration gradient mechanism. Okay, so now in this co-transport, what happens? Molecules are transported across a membrane against their concentration gradients. Okay, by the co-transport of sodium ions, okay, or proton down their concentration gradient. So a carrier protein such as sucrose proton co-transporter, okay, in a plant cell is able to use the diffusion of hydrogen ion down its electrochemical gradient into the cell to drive the uptake of sucrose. Okay, so you can see from here, okay, this sucrose H plus co-transporter, okay, also known as a symporter. Okay, it allow the hydrogen ions or proton to diffuse through this co-transporter. All right, when this sodium ion diffuse, okay, this sucrose H plus co-transporter will take in, okay, take in the sucrose into the cell. So this is a symporter. Symporter means to say it involves two different substances to pass through, not pass through, to be taken in, right, or to be transported by this co-transporter. Two different substances. Okay, uniporter means this co-transporter only allow one substance to pass through or to be taken in or to be pushed out. Now, a proton pump, on the other hand, concentrates hydrogen ions of proton outside the cell. The resulting hydrogen or proton gradient provides potential energy that can be used for active transport of sucrose into the cell. Okay. So class, in this diagram here, all right, it shows you the difference between a co-transporter with a pump. Okay, and this is the proton pump. You can see how it pumps proton across the cell membrane. And since this is a type of active transport, you can see how ATP is needed for the proton pump to pump the proton across the cell membrane. The same goes for the sodium ion pump, the potassium ion pump that we have discussed earlier. ATP is needed. It's just that it is not shown in the diagram, right? Yang tadi itu di sini. Oh, ada. Okay. Alright, penatnya. It's already 48 minutes. Okay, so this is another example. Okay, how a co-transport protein transfer, right, sodium ions and glucose across the membrane. Okay. So you can see from here how the glucose and sodium ion are being transported into the cell. Okay, and you should notice that the concentration of the glucose, right, outside the cell is lower compared to the concentration of the glucose inside the cell. So this is against the concentration gradient. Okay, so you can see there are many other pumps as well. We also have calcium pump, right? Calcium ion pump, not calcium pump, calcium ion pump. So this is the proton pump or hydrogen ion pump, calcium ion pump, sorry, and then sodium potassium ion pump. Okay, and then we have other example, sodium ion pump, potassium ion pump as well. So roughly beginilah how they look like. Okay, so it's sort of like a globular protein. Okay, combination with the alpha helix. Now, bulk transports. Okay, so water and small solids enter and leave the cell by diffusing through the lipid bilayer of the plasma membrane, betul kan? Or by being pumped or moved across the membrane by transport proteins. Now, large molecules, on the other hand, like protein and polysaccharides, as well as larger particles, cross the membrane, okay, cross the plasma membrane in bulk, okay, and packaged in vesicles, okay, either by endocytosis or exocytosis. So these processes require energy. So ini perbezaan antara bulk transport dengan diffusion. Okay, now, like what I mentioned earlier, bulk transport can be endocytosis or exocytosis. Now, okay, so tengok endocytosis dulu. So endocytosis, okay, endocytosis, it is the uptake, okay, you can see uptake of molecules or particulate matter by cell in mass or in bulk by forming vesicle. This is a vesicle. To be more precisely, this is a food vesicle. Okay, some book refer this as food vesicle, some refer as food vacuole. Okay, from the plasma membrane. So you can see the cell, this is the cell. The cell will endocytosize, okay, endocytosize the molecules or particulate matter, okay, and then form a food vesicle. Boleh ya? Endocytosis, endo masuk ke dalam. So kita punya sel makan, okay, sort of like makan lah, okay, makan large molecule itu. Okay. Now divided into one is phagocytosis. Okay, example of endocytosis is phagocytosis. So an example of endocytosis is phagocytosis. All right, so phagocytosis is a process where solid particles taken in by cell. Right, so example is the engulfment of bacteria by phagocytes or macrophage. So a cell engulfs a particle by extending pseudopodia. You can see here. All right, this amoeba is one example of cell. Right, okay, microorganism. Right, that extend its pseudopodia, okay, around the particle. All right, solid particle. To be more specific, solid particle. And then package it within a membranous sac called food vacuole. The particle will be digested after the food vacuole fuses with a lysosome. So meaning to say, the same goes for our macrophage. After our macrophage phagocytosize, okay, phagocytosize bacteria, for example, there are many lysosomes inside the macrophage. The lysosomes will secrete the lysosomes enzymes into the food vesicle, or the bacteria that is already capsulated in a vesicle, okay, and that will digest the bacteria. Now, another example of endocytosis is Pinocytosis. Phagocytosis ini melibatkan solid particles, right? Phagocytosis melibatkan taken in of solid particles into the cell. Now, Pinocytosis, it involves liquid or dissolved substances taken into the cell. Dah beza tau. Okay. Let me rephrase that. Phagocytosis dia melibatkan solid particles dibawa masuk ke dalam sel. Okay. Kalau Pinocytosis dia melibatkan liquid ataupun dissolved substances into the cell. So Pinocytosis is a process where liquid and dissolved substances are taken into the cell in the form of vesicles. Boleh nampak di sini ya. Alright, how these liquid and dissolved substances, they are being taken into the cell. Alright, and then in the form of vesicles. So these vesicles are digested, absorbed, or assimilated by the cell. Okay, so example of Pinocytosis is the uptake of fat droplets in the small intestine. Okay, so a cell continually gulps okay, a cell continually, right, gulps droplets of extracellular fluid into tiny vesicles. Okay, the cell obtains molecules dissolved in the droplets. Parts of the plasma membrane that form vesicles are lined on their cytoplasmic side by a fuzzy layer of coat protein, right? The pits and resulting vesicles are being coated. Now, the third type of endocytosis is receptor mediated endocytosis. Okay, again, the keyword here is endocytosis, so it involves the taken in of substances. But there is an addition here, receptor mediated. So, receptor-mediated endocytosis is a specialized type of pinocytosis. Okay, a specialized type of pinocytosis that enables the cell to acquire bulk quantities of specific substances. Okay. Embedded in the plasma membrane are proteins with receptor sites exposed to extracellular fluid. Okay, now meaning to say, on the surface of our plasma membrane, you know already there are many receptors. Okay, receptors that consist of the glycoprotein. Okay, now specific solutes will bind to this receptor. Right, specific solute will bind to this receptor. Okay, so this receptor protein will cluster in coated pits. Okay, cluster in coated pits to form a vesicle containing the bound of the molecules. So in this example is uptake of cholesterol, right? They are relatively more bound molecules vesicle, but other molecules yang hijau ni, other molecules are also present. Molecules utama yang apa ni endocytosis ni ialah yang purple ni, okay, which is the cholesterol. So after the ingested material is liberated, okay, or freed from the vesicle, the emptied receptors yang Y ni, okay, are recycled to the plasma membrane by the same vesicle. So meaning to say, right, cholesterol ini bind dengan receptor glycoprotein di surface plasma membrane. So what happened? This plasma membrane will form a vesicle, okay. Or to be more specific, okay, okay class, sorry, I guess I'm so tired already. It's 57 minutes already. Okay, so now this receptor mediated endocytosis. So what happens? Okay, let me rephrase. All right, the receptor will bind, this receptor on the surface of the plasma membrane will bind with the solutes, okay, with the solutes, and form a cluster. Okay, this is a cluster. This is a cluster, and then this cluster, okay, will be taken in, okay, taken in a coated pit. This is a coated pit, okay, and this coated pit will eventually form a vesicle, okay, and then in this vesicle, okay, you can see how the receptor bind with the cholesterol, okay. And of course inside this vesicle there are other substances as well. So this cluster, sorry, this vesicle will be ingested by the cell, okay. After this vesicle already ingested by the cell, the receptor, right, which is from the vesicle, remain as it is and will be returned back to the plasma membrane, right, that particular receptor. Now the last one for bulk transport is exocytosis. Now, exocytosis is a process where a cell ejests. Okay, the cell, you can see here, it ejests or secretes substances by the fusion of vesicles with the plasma membrane. So this vesicle from the cell, right, will fuse with the cell plasma membrane and eventually ejects the particles from the vesicles. So secretory cells use exocytosis to export products. Okay, example, amoeba and leukocytes ingested undigested solids from phagocytosis. Right, then cell secretion, where gland cells secretes hormone, then cell in the tubular glands in large intestine secrete mucus, right, is by this exocytosis. Then the cells secretes neurotransmitters such as acetylcholine that signal other neurons or muscle cells. Okay, this acetylcholine you will learn in detail in the second semester. Okay, so this is example of phagocytosis. Okay, and then this is exocytosis. Okay, you can see this vesicle, it moves towards the plasma membrane and ingests the vesicle's contents. Okay, okay, and then this is Pinocytosis. Okay, so that's it. Okay, so 1 hour of today's lecture. Okay, bye.